Good morning, students. Today we will continue with the next production, that is, we are going to start a medical production, that is insulin production. Again and again I am repeating that we have completed lactic acid, citric acid, glutamic acid, aspartic acid. Next, we have completed bread production, SCP, that is single cell protein production. Next, beer, wine. Now today's topic is insulin. Is it okay? Sorry. We have completed penicillin, we have completed streptomycin production, and today we are going to see the 11th production, that is insulin production. Then all of you know what is insulin. What is insulin and where it is produced, everything all of you know this. Now see here, this insulin is a hormone, it is not an enzyme. What is a hormone? Hormone is a chemical. It is a chemical messenger, you can call it as. Hormone is a chemical. It is synthesized at one site and it shows its action at another site. That means enzymes means respiratory enzymes are synthesized and they show their action at one site, that is in the mitochondria. అంటే అంటే మైటోకాండ్రియాలోనే ప్రొడ్యూస్ అవుతాయి, మైటోకాండ్రియాలోనే పనిచేస్తాయి. ఫోటోసింథసిస్, క్లోరోప్లాస్ట్లో ఎంజైమ్స్ క్లోరోప్లాస్ట్లోనే, అంటే ఏంటి, ఎంజైమ్స్ ఏవైతే ఫోటోసింథసిస్లో పనిచేస్తాయో అవి క్లోరోప్లాస్ట్లోనే తయారవుతాయి, క్లోరోప్లాస్ట్లోనే పనిచేస్తాయి. కానీ ఇవి హార్మోన్స్ ఒక చోట తయారయ్యి ఇంకొక చోట పనిచేస్తాయి. సో their site of synthesis and their site of action is different. Such type of chemicals are called as hormones. And then what is the site of synthesis and site of action? Now see here, coming to the insulin. Insulin means insulin is a hormone that maintains the blood sugar levels. All of you know that. Blood glucose levels are maintained with the help of insulin. This insulin is produced in pancreas. See here, this insulin is produced in your pancreas, and when it is produced in pancreas, we have three types of cells: alpha, beta, and gamma or delta. Now, the beta cells of pancreas, the beta cells are also present, that is alpha, beta, and gamma. These cells are present in islets of Langerhans. See here, in the islets of Langerhans of pancreas. See here, the pancreas consists of cells called as islets, special cells called as islets of Langerhans, and these islets of Langerhans consist of three types of cells. Of them, the beta cells of islets of Langerhans of pancreas, they synthesize insulin. Okay? They synthesize insulin. When this insulin is produced, this insulin, it is the site of synthesis. Okay, it is the site of synthesis where beta cells of islets of Langerhans of pancreas is the site of synthesis of insulin. Okay, now, so beta cells of islets of Langerhans of pancreas is the site of synthesis of insulin, and it will show its action in the blood to maintain blood glucose level. It is its site of action. Okay, it is the site of its action. Now, so this is, that means it is having, the insulin is a hormone or insulin is a chemical that is having a site of synthesis and a site of action that are both are present. So it is, this chemical is known as hormone. So the insulin is a chemical consisting of two different sites. One is site of synthesis and site of action. Both are different. So this insulin is known as a hormone. Now, coming to the insulin, how insulin is produced. See here, when you have a heavy meal, మంచిగా ఒక కార్బోహైడ్రేట్ మీల్ లంచ్ టైంలో మంచిగా అన్నం తిన్నాము. when you have a heavy meal, carbohydrate rich meal, immediately that carbohydrate is split into, in the stomach it gets digested in the intestines and long intestines and your intestines, small intestines and large intestines along with stomach it will get digested, and after digestion these large polysaccharides will split into monosaccharides, that is glucose. That means when you have a carbohydrate rich meal, glucose is produced. That glucose level increases, and this glucose enters into the blood. See here, the glucose enters into the blood. When it enters into the blood, immediately what happens? Glucose level increases and it enters into the blood. In the blood also, glucose level has increased. When the glucose level has increased because of this meal, carbohydrate rich meal, glucose enters into our body, and that increased glucose enters into the blood. So in the blood, the glucose level increases. When the glucose level increases in the blood, immediately what happens is, see here, when the glucose level increases in the blood, now insulin also increases in the blood. Insulin level also increases in the blood. When insulin level increases in the blood, then what happens is, what it will do is, this breaks glucose. See here, this converts glucose, I told you wrongly, this glucose is not broken, the glucose is converted to glycogen. And this glycogen gets stored in the tissues. When it is stored in the tissues, see here, glucose level decreases. The glucose level, sugar level decreases, or you can call it as sugar level decreases in the blood, so that your blood sugar level is maintained. This is the course of action, or this is the action of your insulin. See here, when you take a carbohydrate rich meal, immediately glucose level increases in the blood. When the glucose level increases in the blood, along with glucose level, the insulin level also increases in the blood, and this insulin, it converts glucose into glycogen. That increased level of glucose into glycogen, it is the reserve food material of animals, and then this glucose, as we belong to animal kingdom, our reserve food material is glycogen. So this glycogen is stored in the tissues. So the glucose level in the blood is decreased. This is the action of your insulin. Generally, see here, in pancreas, see here, this pre-pro-insulin, okay, the pre-pro-insulin is the initial precursor of insulin. This is synthesized in the beta cells of islets of Langerhans of beta cells of islets of Langerhans of pancreas. This pre-pro-insulin is produced, and it is a precursor. It consists of a signal sequence. It consists of a signal sequence joined to an amino acid, joined to a terminal amino acid. See here, it is joined to a terminal amino acid. Then immediately this will convert into proinsulin. See here, this will convert into proinsulin, and the signal sequence is separated. Instead of signal sequence, 3 disulfide bonds are formed. See here, 3 disulfide bonds are formed in the proinsulin, and it becomes insulin. That means, see here, this pre-proinsulin is the precursor that is synthesized in the pancreas, and it becomes, that is synthesized in the beta cells of islets of Langerhans of pancreas, and this consists of a signal sequence joined to a terminal amino acid. Now it is, as it is a precursor, slowly it synthesizes, it converts into insulin by removing the signal sequence joined at a terminal amino acid, and instead of these amino signal sequence, it produces 3 disulfide bonds. When these 3 disulfide bonds are produced, this proinsulin becomes active, and this starts whenever there is a requirement. When we have food, immediately it converts into insulin and helps in this process. Okay? Now, this is about this thing. Now, what are these 3 disulfide bonds? Now, let us move to our structure of insulin. And coming to the structure of insulin, see here, this insulin structure is coming to the structure of insulin, and this insulin was produced by rDNA technology by Frederick Branty and Charles Branty. This is the production. See here, this insulin structure, it consists of 2 chains, A and B. It is a polypeptide. Insulin is a polypeptide. It consists of 2 chains, 2 polypeptide chains, A and B. See here, this is A and this is B. That means, see here, what? See, this is your, up to here it is A, from here to here it is B. 2 polypeptide chains are present. A chain, see here, I write here, A chain consists of 21 amino acids, B chain consists of 30 amino acids. A chain is made up of 63 base pairs and B chain is made up of 90 base pairs. Okay? A chain is made up of 21 amino acids. See here, 1, 2, 3, 4, 2, 3, 4, 5, 6, 7, 8, or you write it here, 6, 7, 8, 9, 10, 11, like that, 21, 20. I write the main things. Now see here, this is your A chain and B chain. Now see here, B chain 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Like that you will get 19 here. See here, you will get 19. Like that the main numbers are added. See here, insulin is a polypeptide. It is made up of 2 chains, A and B chains. A chain consists of 21 amino acids, B chain consists of 30 amino acids. A chain is made up of 63 base pairs. Its size is, A chain size is 63 base pairs, B chain size is 90 base pairs. Okay? Now, see here. This consists of here, and it is made up, so it consists of 3 disulfide bonds. They are 2 interchain and 1 intrachain disulfide bonds. Okay, it consists of 2 interchain disulfide bonds and 1 intrachain disulfide bond. Now see here, interchain is between 2. Now it is between S, that is A7 sulfur D7. That means between A7 and D7 there is an interchain disulfide bond. Disulfide and 2 sulfurs are present. Next, see here, A20 disulfide bond B90, that is between this a bond is A20 and B19, there is a disulfide bond. A20 and B19, between them a disulfide bond is there. This is known as interchain, 2 interchain disulfide bonds. What is intrachain disulfide bond? Between A6 and A11. See here, A6 a disulfide bond is present A11. This is intra-chain disulfide bond. Between A6 and A11, a disulfide bond is present. Because of these disulfide bonds, its structure is intact and it is A and B chains are not separated. That means total you get, make it total, how many amino acids? 30 plus 21, 51, 105. How much? 6, 5, 156. Generally, people say that there is another chain called C chain. During synthesis, it helps in C chain will help in joining of the A and B chains, but C chain gets separated after synthesis of insulin. So, don't get confused with that. Don't go deep into that. So, we'll see that only A and B chains are present. Now, see here. This is the structure of your insulin. Is it okay, all of you? Now, see here. Its base pairs 90 plus 63. How much? 3, 9 plus 10, 6, 9, 10, 15. See here. 153. Total its size is 153 base pairs. Made up of 51 amino acids. Okay? Made up of 51 amino acids consists of 153. It is made up of 153 base pairs. It is 153 base pairs long. Now, this is your structure. Now, coming to the synthesis of insulin. Is it okay, all of you? Now, coming to the synthesis of insulin. Here what happens is, in the synthesis of insulin, this insulin synthesis the insulin was produced by genetic engineering technique, that is rDNA technology. By means of rDNA technology, this insulin was produced, and this rDNA technology, insulin is produced by Frederick G. Banting and Charles Brett. In 1921, they have produced this recombinant DNA technology, or using genetic engineering. These two scientists, they have produced insulin using E. coli. That is a MicroBial technique. Using microorganisms, they have produced this insulin. Next, this was administered to in 1922, that to January 25th, 1922, they have administered this to a 14-year-old boy. See here, this was administered to a 14-year-old boy who is suffering with tuberculosis, who is suffering with diabetes mellitus, severe diabetes mellitus, that means juvenile diabetes. He is young, so he is having juvenile diabetes. Now, this Frederick G. Banting, he has produced this insulin by using genetic engineering technique from University of Toronto. Where it is situated, University of Toronto means Canada. In Canada, it was produced. Okay? From University of Toronto, this insulin was produced using genetic engineering technology. Coming to the production of insulin, it is a MicroBial production of insulin, and the insulin that is injected to the diabetic patients, human patients, that insulin is named as humulin. See here, it is named as HUMULIN, human insulin. It is known as human insulin. If any of your elders in your house or in your family are taking the insulin injections, you once check the bottle, on that it will be written as humulin, that is human insulin. Okay? Now, coming to the production of insulin, how the insulin is produced. All of you know that take your A chain, take B chain separately. This is 63 base pairs long and this is 90 base pairs long. All of you know that this is your A chain and this is your B chain. Both the chains are taken separately from the insulin. From any person, animal or human being, you separate the insulin and that insulin is taken. Now, these are inserted into P PBR322. Already you have discussed in detail about the structure of PBR322 in your 4th semester. Sorry, in your 5th semester you have studied this PBR322. So, take this PBR322, that is plasmid constructed by Bolivar and Rodriguez, and this is the first ever constructed artificial plasmid. And into this plasmid you will be inserting your A chain. See here, you will be inserting your A chain and take another PBR322 and you have to insert your B chain. See here, you have to insert your B chain. A chain and B chain both are inserted separately into the, these are the A chain is a desired gene and B chain is a desired gene, and these desired genes are inserted into the vector, that is artificial vector PBR322. Into tetracycline resistant marker gene or ampicillin resistant marker gene, you can insert it. When you are inserting it, you have to join ATG. See here, I am joining ATG. What is ATG? ATG is methionine. Triplet codon now that encodes methionine is ATG, adenine thymine guanine. So that ATG triplet codon should be joined to both A and B chains at the terminal positions. Where you have to join? At the point where A and B chains they are joining with each other. At that particular points, after separation from the PBR322 plasmid, after separation from the plasmid, A and B chains will join with each other to produce the entire structure of insulin. Just now we have seen the structure of insulin. Now, these two chains, they will have, you have to join ATG triplet codon sequence, triplet codon that represents, that denotes your methionine should be joined to the end of terminal part of both A and B chain, and these at the point you have to join them so that they can join with each other to construct entire insulin molecule. So it should be joined at the joining site. Now these are inserted into E. coli cells. This is your E. coli cell. Into this E. coli cell you will be inserting your A chain and you will be inserting your B chain consisting of your PBR322. This is also your PBR322. Okay? Now, in this, all of you know that this will multiply, it will replicate and produce many number of copies of A and B chains. Is it okay? Now, when they are multiplying and producing many number of copies, now take these chains, separate them from E. coli cell and separate the A chain from the artificial plasmid, that is PBR322. You will get A chain which is methylated, ATG is present, and B chain is also methylated, that consists of this chain. And when these two are present, see here, now what we have to do is treat these A chain and B chain that are methylated A chain and methylated B chain, you treat it with cyanogen bromide. You treat with cyanogen bromide. When you treat with cyanogen bromide, these methylated sequences are removed. See here, these methylated sequences are removed, and immediately what happens is, see here, this methylated sequence, that is ATG, is removed, and immediately this becomes A, this becomes B. Then what is the necessity of adding and removing it? When you add them, the methylated sequence, its synthesis will be more. That is, insulin synthesis will be more because it is a hormone. Hormone means polypeptide. Polypeptide is made up of proteins or amino acids. So this methionine is the starting codon of protein synthesis. So it helps in the faster replication and production of these insulin A chain and B chains. Molecules of A chain and B chain are polypeptide chains of A polypeptide chain and B polypeptide chain, because it helps in this production, rapid production of A and B chains multiplication, protein synthesis. Now you have to remove with the help of cyanogen bromide. Now these two chains are separated and methylation is removed, then immediately take these A and B chains and you add disulfide bonds. That is, you have to add sulfur. You have to add sulfate so that disulfide bonds are formed between A and B. Once the disulfide bonds are formed, you will produce such type of chain. You will produce such type of chain where between A6 and 11 a bond is formed, that is the SS bond is formed. Between 7 and 7 B and a disulfide bond is formed. 19 and 20 a disulfide bond is formed. And enter complete your this is synthesis. That is insulin structure is synthesized. When insulin structure is synthesized, you have to purify the insulin that is produced in your by this genetic engineering technique using HPLC. That is high performance lipid chromatography. In this, when you release the solution, insulin solution through HPLC, the insulin gets purified. That means purified, or you can call it as pure insulin is produced. Pure insulin is produced. This is known as your production of insulin using genetic engineering technique, that is microbial technique of production of or genetic engineering technique of producing insulin using microorganisms or microbial cells. That is equal cells. This is known as your production. Now coming to the diabetes mellitus. Diabetes mellitus, for treating the patients, you will be giving diabetes injections, and this diabetes mellitus is of what are the symptoms of diabetes? There are two types of diabetes. One is insulin dependent diabetes mellitus, non-insulin dependent diabetes mellitus. What is children? Diabetes mellitus. Diabetes mellitus is the disease. Andara mana sugar diabetes antara dani asal పేరు ఏంటంటే diabetes mellitus. IDDM అంటే insulin dependent diabetes mellitus is also known as juvenile, or it is known as juvenile diabetes. Juvenile diabetes అంటే చిన్న పిల్లలకు వచ్చేది. ఇది చాలా తక్కువ పర్సెంట్, ఇది 10% డయాబెటీస్ mellitus చిన్న పిల్లల్లో వస్తుంది. కానీ ఇన్సులిన్ డిపెండెంట్ వాళ్ళు చిన్నప్పటి నుంచి ఇన్సులిన్ ఇంజెక్షన్స్ ఇస్తారు. నాన్ ఇన్సులిన్ డిపెండెంట్ డయాబెటీస్ mellitus అంటే ఇది 90% పాపులేషన్కి వస్తుంది. ఇది జెనెటిక్ కాంపోజిషన్ అన్న అవ్వచ్చు, లేకపోతే కనుక మన బాడీలో ఉన్న జెనెటిక్ కాంపోజిషన్ అన్న అవ్వచ్చు, లేకపోతే టెన్షన్స్తో కూడా వస్తుంది. నాన్ ఇన్సులిన్ డిపెండెంట్ డయాబెటీస్ mellitus ఇది అబౌవ్ 40 years వరకు, అబౌవ్ 40 years వరకు వస్తుంది. కానీ ఇది మాత్రం 10 years చిన్న పిల్లలకు కూడా వస్తుంది. దిస్ ఈజ్ జెనెటిక్ డయాబెటీస్. అందుకే 14 years old బాయ్ ఫస్ట్ టైం January 1922లో 14 years బాయ్కి జెనెటిక్ డయాబెటీస్ ఉంది కాబట్టి ఈ సఫరింగ్ IDDM, దట్ ఈస్ ఇన్సులిన్ డిపెండెంట్ డయాబెటీస్ mellitus కాబట్టి, ఆర్ జెనెటిక్ డయాబెటీస్ కాబట్టి ఆయన 10% వస్తాడు కాబట్టి ఆ బాబుకి ఆ అబ్బాయికి ఈ ఇంజెక్షన్ చేశారు. ఓకే, నౌ this diabetes mellitus, what are the symptoms of diabetes mellitus? The symptoms of diabetes mellitus is that here, sorry, I missed this one. When these two, when they join with each other, this is known as pro-insulin. This is known as pro-insulin. To this we are joined disulfide bonds and it has converted into insulin. Now this is your insulin. Is it okay? Now, the symptoms of the insulin diabetes patients are polyuria. They will be urinating a lot. Chala salu vaattuntaru. They are thirsty and regularly they will drink large amount of water. They will be drinking large amount of water. Polyuria, drinking will be more, then your sweating will be more. Okay, they will be releasing sweet urine, that is in the urine also the glucose will be coming out. That is known as sweet urine. And they will become very weak and they show weight loss. These are the symptoms of your diabetes patients. So with this today we will stop here. the insulin production, and in the next class we will continue with the monoclonal antibody production. Thank you, students.